video on the drug chlorphenamine and will inform you as a pharmacist of everything you need to know to treat with chlorphenamine. Firstly, it's important to know that chlorphenamine, which can also be known as chlorphenamine, is a first-generation antihistamine class of drug that is used to alleviate the symptoms of allergic disease. Allergy affects many people within the population. For example, hay fever is estimated to affect over 10 million people in England alone, according to the NHS Choices. We will now go into detail about what allergy is and how it affects its sufferers.
Allergy is an unnecessary immune response to a non-harmful foreign substance. We currently do not know what causes them, but we know that an early introduction of the allergens does decrease likelihood of developing an allergy. It begins with sensation where a small amount of IgE is produced and the T cells then recognize the allergen. So when the allergen is reintroduced, a massive amount of IgE is produced, which in turn causes the allergic response. Firstly, the T lymphocytes identify the allergen that has been introduced. Once this occurs, the T lymphocytes transform into plasma cells, which secrete the IgE antibodies. These IgE antibodies then go on to bind to mast cells in tissue and basophils in circulation. The IgE binds to the Fc epsilon receptor. This binding triggers the histamine that was restored in granules inside the mast cells and basophils to be released into the bloodstream and lymph. Mast cells are mostly found in connective tissues and are responsible for storing, synthesizing and releasing of chemical messengers to the local tissues. Basophils are the same as mast cells, however are produced in bone marrow and are in circulation rather than being stationary in a certain tissue. Histamine is synthesized in the cells by L-histidine decarboxylase, which converts histidine into histamine. This is the molecule in question. Histamine is a simple molecule with an imidazole ring with a hydrophobic chain and a primary amine. When in contact with the H1 receptor, the receptor that triggers allergic effects, it binds by bonding at specific sites. One of the imidazole nitrogen donate a lone pair to the lysine at position 200. The other nitrogen in the imidazole ring donates its hydrogen to bind to the asparagine at position 207. The primary amine donates both its hydrogens to form two hydrogen bonds with the aspartic acid at position 116. The H1 receptor is a G-protein coupled receptor. Once the histamine binds, it triggers the internal cell signaling pathway. This pathway causes an opening of calcium 2+ channels, causing a surge in the concentration of calcium 2+. If the channels are blocked, then the cell will use its internal calcium store. This increased calcium concentration causes cell effects. For example, in nerve cells it causes impulses which constantly fire, causing itching. In epithelial cells, especially in the nasal cavity, it causes increased mucus production. Most commonly, allergy causes symptoms such as sneezing, an itchy or runny nose, itchy red watery eyes, wheezing, shortness of breath, chest tightness, raised itchy red skin, swollen lips, tongue, eyes or face, dry red cracked skin, stomach pain, or nausea and vomiting.
Chlorphenamine is often given as chlorphenamine maleate, as seen on the right-hand side of this slide. As an antihistamine drug, chlorphenamine is a H1 receptor antagonist. The structure of the alkyl amine is rather simple, with just three major functional groups, and obeys Lipinski's rules of oral availability. As a competitive antagonist, chlorphenamine competes with histamine for the receptor site on the G-protein coupled H1 receptor on effector cells of the gastrointestinal tract, blood vessels, and respiratory tract. Chlorphenamine provides effective temporary relief of sneezing, watery and itchy eyes, and runny nose, which are symptoms caused by hay fever and other upper respiratory allergies. The indications for treatment with chlorphenamine include the symptomatic relief of allergies such as hay fever, urticaria, food allergies, and drug reactions. It can also be used to relieve the itchiness associated with chickenpox. As mentioned, histamine receptors are G-protein coupled receptors, meaning that they effectively act as cellular switches, which can exist in either the active or inactive state, dependent on cross-linking between different domains of the receptor. Histamine interacts with domains 3 and 5. The protonated amine interacts with aspartate residues in domain 3 and uses this as a counterion. The imidazole ring forms hydrogen bonds with asparagine and lysine residues in domain 5. H1 antagonists are nitrogenous bases containing an aliphatic side chain, sharing with histamine the common core structure of a substituted ethylamine. The ethylamine side chain is essential for H1 antagonism. Different substitutions, for example, a methyl group in the 2 position, can increase binding affinity. This plays a key role in the development of chlorphenamine, which will be talked about later in this video. Some features are key in binding to the H1 receptor, such as the presence of multiple aromatic or heterocyclic rings and alkyl substituents, and the basicity of the nitrogen group. The effects come primarily from inhibition of action at H1 receptors, which temporarily stops histamine binding and causing typical allergic symptoms. It doesn't act to reverse these effects, but acts as a simple antagonist and blocks them. First-generation antihistamines can be displaced by high levels of histamine. There is some evidence to suggest that antihistamines may be able to block mediator release from basophils and mast cells, which has led to speculation that chlorphenamine could also act as an inverse agonist. Moving on to the side effects of chlorphenamine, the first-generation antihistamine has many downfalls. Off-target side effects are caused by chlorphenamine binding to receptors other than the H1 receptors, and this is due to its poor selectivity for the H1 receptor. Chlorphenamine can bind to cholinergic muscarinic, alpha-adrenergic, and serotonin receptors and have adverse effects by these mechanisms. Due to chlorphenamine's very poor selectivity, it has very many off-target side effects. These include the blocking of serotonin receptors in the brain, causing depression. Blocking of cholinergic muscarinic receptors can cause antimuscarinic effects such as dry mouth. Blocking of alpha-adrenergic receptors, slowing the heart rate and causing hypertension, blocking of cholinergic receptors causing blurred vision, and blocking of cholinergic receptors leading to prolonged QT and hence cardiac arrhythmias. On-target side effects are caused by binding directly to the H1 receptors in the body. Histamine is an important neuromediator in the brain, and stimulation of the H1 receptor in major parts of the cerebrum, cerebellum, and posterior pituitary increase arousal in the circadian sleep-wake cycle and reinforce learning and memory. Hence, blocking of these H1 receptors can cause CNS depression, which will most often present as drowsiness and mind fog, as well as fatigue. This is all down to chlorphenamine's ability to cross the blood-brain barrier, owing to its lipophilicity and log p-value of 3.8.
The history of the discovery of chlorphenamine and first-generation antihistamines begins with the discovery of histamine in 1910. In 1937, Bovet synthesized piperoxan from acetylcholine, but it was not effective in diminishing histamine's action. Fenbenzamine belongs in the ethylene diamines group. It was the first drug suitable for clinical use. In triphenamine, when the benzene was replaced with a pyridine, the potency increased. And later, in meperamine, parasubstitution with the lipophilic methoxy group increased the potency and lipophilicity, and also decreased the toxicity. Since all these are lipophilic, they can cross the blood-brain barrier and thus have sedative effects. In 1943, diphenhydramine of the aminoalkyl ethers group was synthesized. The connecting atom changed into a carbon connected to an oxygen. Bromodiphenhydramine was twice as potent because of the parasubstitution of bromine. Similar compounds were synthesized. For example, in carbinoxamine, benzene was replaced with a pyridine and also a para-chlorosubstitution on the ring, which massively increased the potency. Chloride increased the lipophilicity also and metabolic stability. In 1945, phenethazine was the first member of the phenothiazines group. It had 2 nitrogens. Both were basic, but the second one was more basic due to the less steric hindrance and the absence of withdrawing groups around it. Moving on to propylamines, in 1948 a series of long-lasting antihistamines was synthesized, with phenyramine being the prototype and the weakest member of the 3. The connective atom in this group is a carbon. In chlorphenamine, halogenation at the para position increased the potency 10 to 20 times more, with no significant increase in toxicity.
Hi, today I'm going to show you how the chlorphenamine is synthesized. So here's the general chemistry of the chlorphenamine. Here is the chlorphen acetyl nitrile. And the sodium amide here works as a very basic nucleophile. The lone pair electrons in the amine just attack the hydrogen in this way. And then this is the deprotonation. And it gives a structure like this, with a negative charge here, and then it attacks and get rid of the halogen here, and then the progress just happen just similar way in this way. And then we finally got a H2SO4 here for the decarboxylation, and then we can get a chlorphenamine here. So this is the first method to synthesize the chlorphenamine, and there's another method where it can synthesize the same thing as well. However, the main difference is we use the pyridine instead of the chlorpyridine in here. The synthesis route 1 is more preferred than the synthesis 2 because the halogen here is a very good leaving group, and it can make the reaction more likely to be happen.
So talking about absorption, it's absorbed slowly from the gastrointestinal tract, and the peak plasma concentrations occur about 2.5 to 6 hours after oral dose. About 70% of chlorphenamine in circulation is bound to plasma proteins, and there's a wide range of inter-individual variation of the distribution of chlorphenamine. Values ranging from 2 to 43 hours have been reported for the half-life. It is increased in the presence of renal dysfunction and decreased in children. Chlorphenamine is metabolized predominantly in the liver via the CYP450 enzymes, and it is also metabolized in the lungs and the kidneys. Chlorphenamine undergoes enzymatic hydrolysis, and the bioavailability of oral solution and tablets are 34% and 25% respectively.